زمان ما شفتكم إلنا سنة. أوكي. بالموديول هذا كان الدكتور نايف كراتشي يدرسه على 8 محاضرات. I think إنه أنا راح أكون معاكم 6 محاضرات. لا 5. وفي واحدة راح تنزل أونلاين إن شاء الله على اليوتيوب لكم. دنس عبارة عن محاضرتين. فعندنا total of 7 lectures. what I'll do راح نتكلم اليوم إن شاء الله introduction يعني introduction و refreshment للميموري تبعتكم على الهيموجلوبين. طيب؟ وراح أضيف أشياء جديدة على اللي درستهم إياه قبل سنتين. وبكرة راح نتكلم عن regulation of hemoglobin. راح نتكلم المحاضرتين اللي نزلوا وراح ينزلوا لكم على hemoglobinopathies, diseases, disorders related للهيموجلوبين molecule. بعدين عندنا metabolism in erythrocytes وعندنا metabolism of iron اللي هو basically absorption, storage, transport, genetic diseases related to iron. And we have a final lecture on coagulation. From a biochemical point of view, يعني في overlap حيكون بين الفسيولوجي والبايوكيمستري، بس from a biochemical point of view راح نتكلم عن coagulation إن شاء الله. هذه in the 3rd week. So, what I'll do now is go over some basic information about hemoglobin. If you have any questions, I'm assuming that you remember. Okay? If my assumption is wrong, just raise your hand. Please let me know. واحكوا لي احنا نسينا هذا الحكي. هلا. So let's go over the first topic. So let's start with heme. You studied heme in the metabolism in biochemistry too. Heme is a molecule that is made of protoporphyrin 9 plus iron. So this is protoporphyrin 9 right here. Okay? And when iron is associated with it, it becomes heme. Now, this protoporphyrin, or the heme molecule, which looks like this, is a planar molecule. It's a flat molecule. And it has associated with it 3 groups. These are the phenyl, the methyl, and we have the propionate group. Now these groups are, these 2 groups are hydrophobic. And if you look at this whole molecule, it really looks, it's hydrophobic. It's a hydrophobic molecule, except for these 2 groups, the propionate group that is hydrophilic and it is charged. And this has an important implication when it comes to how heme interacts with hemoglobin. And it does have important implication when it comes to hemoglobinopathies. So the heme molecule is made of 4 chiral rings. That's a chiral ring. So you have 1, 2, 3, 4. So they're designated as A, B, C, and D. In the middle you have iron. And iron can form 6 covalent bonds. We call them 6 coordinates. 4 of them exist between iron and the chiral and each chiral ring. Okay? And we have 2 other ones. One with histidine, the 5th coordinate, and the 6th coordinate is with oxygen. Now, heme is important in the function of many proteins, collectively known as hemoproteins. And heme is known as a prosthetic group. A prosthetic group is a non-protein group that interacts covalently with a protein. Okay? So there are different heme proteins or hemoproteins. Probably the most well-studied and common and famous are myoglobin and hemoglobin. And they have their dysfunction in binding to oxygen. But you have other ones. You have proteins like cytochrome P450 enzymes, which exist in liver, and they are important for detoxification and metabolism as well. But you have also heme as part of cytochrome proteins. and they are important in electron transport, in the electron transport chain, metabolism. But heme can also exist in other gas sensors or gas sensing proteins. They sense, these proteins that sense if there is carbon monoxide, oxygen, nitric oxide, and so on. So that's heme, that's the importance of heme. You studied heme biosynthesis before, but I'm gonna refresh your memories and ask you to learn this all over again. And this is important for you in the future, as well as important for the other sections in this module. So basically, heme synthesis starts with the integration of two molecules, glycine and succinyl coenzyme A. This is the rate-limiting and committed step. Rate-limiting means that it is the slowest in a pathway. Committed means that there's no point of return. It doesn't go back. It has to move forward and finish off what it has started. So, resulting in the formation of a molecule known as aminolevulinic acid, or ALA. ALA continues through different intermediates, forming at the end protoporphyrin, where it integrates with iron, forming heme. Now, this very last step occurs spontaneously by itself, but you can have an enzyme as well that is necessary that catalyzes this reaction, and is known as ferrochelatase. The first reaction requires vitamin B6, and it is regulated by hemin. Hemin is the heme molecule that has iron in the ferric state, that is Fe plus 3. Okay, so the heme that we're talking about is, it has a ferrous iron, or iron in the ferrous state. Okay? Questions? نعم.
هلا آخر ستيب إيش هو الإنزيم اللي بيخليها تتسرع أو accelerate؟
آخر step, ferrochelatase. That's the name of the enzyme. Now the first reaction and the last one can be inhibited by lead as well. Now this is the sequence of synthesis of heme, as well as disorders related to heme biosynthesis. I want you to know the names of these disorders. I'm sure you studied them before in Biochem 2. I want you to know the names. I want you to know the genetic pattern of disorders, whether they are autosomal recessive or dominant. I want you to know the enzyme responsible for that, and I want you to know if the clinical manifestation, particularly or specifically, whether the patient having the disease is photosensitive or not. Now, photosensitivity is related to the formation of the pyrrole rings, of having these pyrrole rings. And it starts with this molecule, hydroxymethylbutane. Okay? So any disorder that results from enzymes, from defective enzymes early on, patients would be photoinsensitive. That is, they are not affected by sunlight. But any disorder, any defect in an enzyme that takes place afterwards, after hydroxymethylbutane, then patients would be photosensitive. The main organs that are affected is the nervous system as well as the digestive system. For certain reasons, patients would have cramps and stomach pain. Now, in addition, the organ that is affected could be liver or the erythroid tissue. Okay? Synthesis of heme occurs in both erythrocytes or erythroid tissue plus liver. So these are the main two organs that can be directly affected by any mutation in any of these enzymes. The predominant porphyria, porphyria is the disorder that is related to synthesis of heme. So the most common porphyria is known as cutaneous tarda, which is right here, this one right here. Okay, porphyria cutaneous tartar. This is the predominant, about 80% of porphyria is of this type. Porphyria, by the way, is a Greek word that means purple. And the reason why they called it this way is because the urine of patients would have this purple color. Okay? And this is the structure of the hydroxymethylbutane, that is the first one that has this ring structure, the pyrrole ring structure, the 4 pyrrole rings. Treatment, briefly, patients are given hemin. Why? Because hemin is actually the inhibitor of the very first reaction way. Okay, so they're given intravenous injection of hemin so that they would not produce a lot of any of these intermediates. Okay, that would accumulate as a result of defective enzymes. Also, patients are protected from sunlight for obvious reasons, because they would have lesions on their skin as a result of accumulating these intermediates, and they are given, or they're given a lot of antioxidants, specifically beta-carotene. The reason is that these intermediates would result in the production of oxygen radicals, and they must be removed by these antioxidizing agents, antioxidants like beta-carotene. Okay? Questions? So let's move on and talk about hemoglobin. Hemoglobin is a globular protein. Remember, we talked about classification of proteins according to structure. We have 2 types, fibrous and globular. Globular proteins, like hemoglobin, are characterized by having the polar charged amino acids on the surface, and they have the hydrophobic non-polar amino acids in the core, in the middle. Okay? So this is the basic structure of hemoglobin. Now, except for, there's an exception, which is in the center, in the core of hemoglobin, we have 2 histidines, which are positively charged. And these histidines are important for binding to heme and stabilizing binding of oxygen to heme. So right here you have the proximal histidine that binds covalently to iron in the middle of heme. And when oxygen comes in, and it interacts with the iron in the 6th coordinate, this interaction is stabilized by the other histidine, which is known as the distal histidine. In the lecture where we discuss hemoglobinopathies, pay attention to mutations that can take place in either one of these histidines, the proximal or the distal. Because you can have a mutation in this histidine that can affect binding affinity of oxygen to hemoglobin. Okay? Now, hemoglobin is also an allosteric protein. Allosteric means different shape. So allosteric proteins are proteins that have different shapes or different structures. These proteins are characterized, allosteric proteins are characterized by having multiple subunits, multiple polypeptides. So they are not monomeric like myoglobin. No, they are multimeric. They can be dimers, trimers, whatever. So so hemoglobin is made of 4 polypeptide chains. In adults, the predominant hemoglobin is made of 2 chains, 2 alpha chains and 2 beta chains. Now, each one of them has a heme bound to it, and each heme can bind to an oxygen molecule. So each hemoglobin molecule can bind to 4 oxygen molecules. Now, the other thing is that this hemoglobin, as an allosteric protein, it has different structures. And there are 2 structures, the R state and the T state. R means relaxed. This is the structure of hemoglobin that makes it having high affinity for oxygen. So R state is the high affinity state. The T state, where T stands for tense or tau, means that this hemoglobin molecule has low affinity for oxygen. Okay, so there are two structures, R high affinity and T low affinity. Now there is also positive cooperativity. As an allosteric enzyme, as an allosteric protein, it has cooperativity. And this cooperativity is positive for oxygen. What it means is that binding of one oxygen makes it easier for a second oxygen molecule to bind, which makes it easier for a third one to bind, and so on. Okay? So that's the meaning of cooperativity. Now the other thing is that, as an allosteric protein, it can be regulated by effectors, other molecules. And these are known as allosteric effectors. And these allosteric effectors can be positive or negative. And these allosteric effectors can be homotropic or heterotropic. We'll talk about this in a second. So the question is now, okay, so hemoglobin is a tetrameric protein. It is a tetrameric protein. How does it form? So I came up with this term. It's a dimer of a dimer, which means what? It means that it's actually, you have two dimers. Each one of them is made of alpha and beta. And the other one is also alpha and beta. And these two dimers coming together, forming a tetramer. So in each one of them, you have two alphas and two betas. First, if you take one dimer, how do these two polypeptide chains, the alpha and beta, how do they interact with each other? They interact with each other via hydrophobic interactions. So yes, I said before that hydrophobic amino acids exist in the middle, in the center, in the core of a protein. But yes, for hemoglobin, you can have some non-polar hydrophobic amino acids on the surface. And these hydrophobic amino acids are important for the formation of the alpha and beta dimer. Now these two dimers, alpha and beta, they interact with each other via electrostatic interactions. So right here you have hydrophobic interactions, but the interaction between this dimer with that dimer is mediated by electrostatic interactions as well as hydrogen bonds. And these are really important in changing the structure of hemoglobin from R to T and T to R, as we will talk about. I'll show you in a second. All right. Something else about how heme binds or interacts with hemoglobin. Remember how I said that heme is mainly hydrophobic except these two propionate groups? Well, this is how heme fits into the hemoglobin molecule. Here, right here, this is the hydrophobic part of heme. It's embedded inside the hemoglobin molecule, where you have this interaction with the proximal histidine, and it's also nearby the distal histidine. The propionate structures are on top. They are oriented outward, interacting with the polar amino acids on the surface of the protein. So the heme molecule, the interaction of heme molecule with hemoglobin is stabilized by two forces: the hydrophobic interactions inside, as well as the hydrophilic electrostatic interactions with the propionate structures. This has really important implications when it comes to hemoglobinopathies. That is, changing the hydrophobic environment that surrounds heme can really affect this interaction between heme and the protein, and it can also affect oxygen affinity towards hemoglobin. Okay? Questions? أمور تمام؟ ذاكرين هذا الحكي ولا ناسينه؟ Let's move on. So what is the function of hemoglobin? Transporting oxygen. So if you look at it, now inspired air, look at the pressure of oxygen, how much oxygen there is in air that we inspire. It's about 160, 150, 160 torr, millimeter of mercury. When it goes into the lungs, it's about 104. When it leaves lungs, it's about 100. torr. So this is, high level of oxygen bound to hemoglobin. And hemoglobin would be almost 100% saturated with oxygen, bound with 4 molecules of oxygen. Now, as it goes into peripheral tissues, notice that the level of oxygen goes down to about 40 torr. This is where and when hemoglobin releases oxygen to peripheral tissues. Now, when it goes back to lungs, now it has low level of oxygen. It's hardly saturated with oxygen. It's empty. Well, it doesn't carry as many oxygen molecules as before. It's about 40 torr. So we start with about 100 coming out of lungs. When it goes to tissues, it becomes, the pressure of oxygen is about 40 and oxygen is released. Now, this is why, because of this, what oxygen binding does to hemoglobin is that it gives it this red color. So when hemoglobin does not have oxygen, now hemoglobin has this blue color to it. Fine. So this is the oxygen saturation curve. The idea is that as an allostatic protein, it has a sigmoidal plot. It looks sigmoidal, meaning that you have this transition in the structure. It's not hyperbolic like myoglobin, if you remember. So it has this S-looking structure. Now, this structure indicates that this protein has 2 structures. The shape of this plot indicates that the protein has 2 structures. A structure right here indicating that this protein has low affinity for oxygen, and another structure that is right here, it exists right here, where it means that the protein has high affinity. So these are the 2 structures. The T state right here and the R state up there. So it does make sense. Now we want hemoglobin to be in the R state in lungs so that it can bind to as many oxygen molecules as possible, to be saturated. And we want it to have low affinity when hemoglobin reaches tissues so that it releases this oxygen to peripheral tissues. So you have a transition from low affinity state to the high affinity state. Now notice that here at rest, when you're just sitting like a couch potato watching TV, the pressure of oxygen in peripheral tissues is about 40 torr. But when you're exercising, notice that when it goes down to about 20, look at the drop. Look at the drop of oxygen saturation from about 80% all the way to about 40%. Huge drop in release, in saturation of oxygen. At high altitudes, when the pressure of oxygen goes down, it's about 5,000 meters above sea level, the amount of alveolar oxygen is about 75. So about 75, which is right here, you have the level of saturation of hemoglobin is about 90%. Still, hemoglobin is mainly saturated, which is good. This is not for you to memorize, but to understand. So at sea level, like we're here right now, the atmospheric pressure is about 760. The arterial pressure of oxygen is about 100. Now, venous pressure of oxygen is about 40. So the level of saturation right here is about almost 98%, almost 100%. Okay? So the amount of oxygen released is about 60 torr. Now, if you go up high to about 10,000 feet, that is about 3,000 meters, notice how the level of saturation is about 90%. Look at this plot right here. I love this. This is at sea level, right here. This is inspired air. This is how much oxygen you breathe in. It's about 150. Okay. Now, if you go high in altitudes, that's in meters. So if you go high in altitudes, the highest human habitation is about 6,000. This is Mount Everest right here. No one lives there. People would have a really hard time breathing because there is hardly any oxygen. Oh, there is oxygen, but hemoglobin is not saturated. Okay? Actually, I don't know if you've ever experienced it or not, but I did. I went to this level right here, almost this level right here. And I was really having a hard time breathing. It's amazing. Anyhow, so the question is, how does this change take place? In other words, what does happen to hemoglobin structure? How does it go, this transformation, this transition from T to R or R to T? Going back to basic biochemistry, it's all about little structural changes. So what happens is heme is a flat molecule, but when it sits inside hemoglobin, it is not flat. It looks like a dome, زي القبة. It looks like a dome. Why? Right here, you have the proximal histidine. It's bound to iron. This is the fifth coordinate. On top, right here, you have the distal histidine. Since heme is hydrophobic, there is repulsion between the hydrophobic heme with the charged histidine. So there's repulsion. So what happens is that heme sort of like bends a little bit so that it has this structure. With iron outside the plane of heme. It's outside, so it's not sitting exactly in the middle because of this repulsion. But when oxygen binds, this repulsion is reduced, and rather you have this nice interaction between histidine, the distal histidine, with oxygen. So there's a change in the structure of heme from this dome-like structure to a flat structure, and iron now is sitting in the middle, in the center of the heme molecule. There is a little change right here in the structure of heme, and this change is about 1 nanometer. Very little, minute change in the structure of heme. But this change has huge consequences, because what happens is that when heme moves from this structure to a flat structure, it pulls proximal histidine a little upward. That changes the interaction between histidine with the amino acids that come afterwards in the alpha helix, because now it is sitting in an alpha helix right here. That changes the structure of the alpha helix, that is changed in the secondary structure. This also changes the tertiary structure of the molecule, and that changes the overall quaternary structure of hemoglobin. Okay? Now, so what happens is that this sequential change in the structure of hemoglobin, it leads to change in the quaternary structure. But what does exactly happen? Basically what happens is that you have a break in the electrostatic interactions. So when you have this, when oxygen binds to hemoglobin, the number of electrostatic interactions between these two dimers is reduced. Electrostatic interactions are broken off. So the molecule becomes relaxed because there are less interactions than before. So that changes the T state, the T structure, to the R structure, to the relaxed structure, the high affinity structure. So it's all about electrostatic interactions that take place in the middle, in the center of the hemoglobin molecule. So we're talking about changes that take place right here in the center. Okay? When you have less electrostatic interactions in here, that leads to this transition from T to R. Okay? Questions? I know انكم معتمدين على الفيديو انكم تطلعوا عليه يعني. بس you can ask questions please يعني عشان. Yes.
بس لما اخذنا على البي 50 ممكن تتغير بالهاي altitude ولا لا؟
No, the P50 stays the same. Ah, wait. لا, the P50 will change actually. Tomorrow when we talk about regulation of hemoglobin, and we will talk about how people adapt. In high altitude, they do adapt. They do have different P50. We'll talk about that tomorrow, inshallah. Yes, sir. ما فهمتش علي ايش؟ don't like? Okay, ماشي ماشي. بس في سؤال طيب ماشي. Okay. So basically the idea انه انت عندك الهيم, it looks like this. هلا it should look like flat, but it doesn't look flat. It looks like a dome, like this. ليش؟ لأنه you have this because you have distal histidine on top, okay? Creating a repulsion between the histidine with the iron in the middle. So iron is pushed downward. So heme looks like this, okay? So it looks like a dome. هلا, and you have this interaction, covalent interaction with the proximal histidine. Okay? Now what happens when oxygen comes in and it binds to iron, there is no repulsion, no more, there is less repulsion. There is good interaction, favorable interaction between oxygen with the distal histidine. So iron is pushed upward, okay? It becomes flat. The heme molecule becomes flat. Now, it looks like this. When it comes flat, this histidine, the proximal histidine, is pushed upward, pulling the amino acids that are linked to it by peptide bond, changing the alpha helix. So right here, عندك change من الدوم بيطلع لفوق شوي. That pulls the amino acids right here that comes that are interacting with the histidine. It changes the structure of the alpha helix. The whole alpha helix is pulled up as well. That changes the tertiary structure and the quaternary structure. Okay? Yani if you remember 2 years ago when I told you that you have oxygen comes in, it changes the structure a little bit. Yani if you rotate this finger, it rotates everything with it. Everything that interacts with it. Eventually changing the whole structure of the molecule. Okay? Now, so electrostatic interactions. And binding of oxygen to hemoglobin is cooperative. Meaning what? Meaning that when one oxygen binds to one of these chains, one heme, it makes it easier for a second one to bind. It makes it easier for a third one to bind. So there is increasing affinity towards oxygen. And this is the meaning of cooperativity. And that is why you have this sigmoidal shape. That's why you have this sigmoidal shape right here, because it becomes more and more, it becomes easier and easier for another molecule of oxygen to bind. So right here you have the T state, and right here you have the high affinity R state. Okay? Hello. So let's go back showing you a basic biochemistry. So biochemists said, wait a second, what does happen exactly to the hemoglobin molecule? Yani, what does really happen to hemoglobin with and without oxygen? So they came up with a number of hypotheses. I'm gonna focus on 2 of them. The first one, known as the concerted model. It's also known as the MWC model. MWC, these are the initials for the 3 scientists who came up with this hypothesis or this model. They said the following. They said that hemoglobin exists in 2 structures only. And these structures are the T state and the R state. And these 2 structures exist in equilibrium. Okay? So for example, if you have a hemoglobin molecule without oxygen whatsoever, and you look at these hemoglobin molecules, you would see that 95% of them exist in the T state. And 5% of them exist in the R state. And Close your eyes, open your eyes, look at them again, 95% and 5%. So there's equilibrium. It's not the same molecule that stays in the R state. No. The molecule changes structures R to T. But it stays in the T for a long time. It can change to R. Okay? But really quickly it changes back to T because there is no oxygen. Got it? So there are two structures only, T and R, and they exist in equilibrium. If there is no oxygen, you look at hemoglobin molecules, let's say you have 100 hemoglobin molecules, 95 of them, 95% that is, they exist in the T state, and 5% exist in the R state, all the time. Okay? If you add one oxygen molecule, it binds to one of these heats. Changing the equilibrium, shifting the equilibrium towards the R state. So if a hemoglobin molecule has one oxygen bound to it, there is a chance, there's a higher probability that it would exist in the R state, not in the T state. So if you add little oxygen to an environment, to a chamber that has hemoglobin molecules, you would notice that 75% of them exist in the T state and 25% exist in the R state. Okay? Now, increase oxygen a little bit. Now, the equilibrium shifts even further. Now, notice here, you have only two structures, T and R, and you have an equilibrium. There is no oxygen right here. Mainly, the arrow goes towards the T state. So hemoglobin exists in the T state more than existing in the R state. If you add little oxygen right here, notice the length of these arrows changes. Now, yes, hemoglobin exists in the T state, at a higher probability, at a higher ratio, but there is higher proportion, higher ratio, or higher amount of hemoglobin in the R state. Increase oxygen a little bit more. Now the equilibrium also shifts towards the R state. Increase it more. Eventually, if you have hemoglobin, all chains bound to oxygen, it mainly exists in the R state, not the T state. Okay? So that's basically what it tells us. It tells us that 1, hemoglobin exists in two states, T and R. 2, these two states exist in equilibrium. 3, oxygen changes or shifts the equilibrium towards the R state. Okay? These are the 3 important points. Now, oxygen is known as an allosteric homotropic effector. Okay? So it is an allosteric effector. Why? Because it changes or it regulates the allosteric protein. So it is an allosteric effector. Homotropic means that it changes the affinity for binding of another oxygen molecule, another molecule of the same type. That's why it's known as homo. Okay? But you do have some other heterotropic effectors. We'll talk about them tomorrow. Like carbon dioxide, carbon monoxide, bisphosphoglycerate. We'll talk about them tomorrow, inshallah. Okay, so these are known as heterotropic allosteric effectors. They are allosteric effectors, they affect an allosteric protein, and they are heterotropic, meaning that they are of a different type than the ligand oxygen that hemoglobin binds to. Now,
بالتالي يعني الاكسجين حسب هاي النظريه بيكون allosteric effect مش بس للهيموجلوبين اللي هي بتوصل فيه لأذر هيموجلوبين.
No, the hemoglobin itself. The hemoglobin, the cell molecule itself, yes. Okay. So, it turns out that this model works perfectly fine for hemoglobin. Okay? لأنه hemoglobin really exists in two states, T and R. And it explains the effect of oxygen really well. But it does not explain the effect of the other allosteric modifiers or allosteric effectors. Scientists came up with another model, and they called it the sequential model. Sequential means what? It means in sequence. It's also known as the induced fit model. Remember induced fit model of enzymes, how the active site changes a little bit so that it can fit the substrate? Okay, so the same thing here. That is, you have a little change in the structure of the hemoglobin molecule to make it easier for a second molecule to bind. It's also known as the K and F model. These are the initials of the 3 scientists who came up with this model. So the idea behind this model is the following. Hemoglobin exists in T state and R state, and also in other structures in between. So what happens here is that when an oxygen molecule binds to one of these chains, it changes the structure of this polypeptide, and it changes the structure of the neighboring chains just a little bit. When another molecule binds, it also changes the other chains, and so on and so forth, until you get the structure right here where you have a fully R state. Okay? So sequentially, the structure of hemoglobin changes, and you have different structures. Not only that you have T and R, but you have also structures in between these 2 structures, the T and the R structures. And this model system, it turns out to be a bit less accurate than the previous one, but it turns out that it explains the effect of the heterotropic effectors better than the previous one, without going into details. Then it's all about mathematical equations and experiments and all that. But that's all I want you to know. Okay? So these are the 2 models. Now, questions?
So,
From
in the conservative model, multiple configuration change.
Yes, Hala. I forgot. Thank you for this question, and I forgot to mention something that's really important. It turns out, Barlo, that both models are accurate, but the conservative model explains things better. But both are accurate, and the scientists discovered, like I said, that you do have changes in the tertiary structure. In this model right here, changes occur at the quaternary structure. You have R and T, right? The quaternary structure. This model right here says, no. Before having this quaternary structure, we also have tertiary changes in the tertiary structures within the same polypeptide, and it turns out to be true. Yes. So scientists said, it's not about quaternary structure altogether at the end. No, you do have some changes in the secondary structure and tertiary structures as well. But mathematically and experimentally, the conservative model works best. But we keep talking about hemoglobin as an alpha-beta protein, alpha-2, beta-2 protein. But there's more than that. There are more hemoglobin molecules than that. We have hemoglobin molecules that exist in the embryonic stage. We have ones that exist in the fetal stage, and we have the adult hemoglobin that we know now, the alpha and beta, alpha-2, beta-2. So, now, in blue you have the alpha-similar globin proteins. In red you have the beta-globin proteins. You have to have the right ratio between alpha and beta. Okay? So, initially in the embryonic stage, from the yolk sac, you have production of two chains. You have the presence of 2 chains, expression of 2 genes. These are known as globin zeta and globin epsilon. So in the embryonic stage, you have a hemoglobin molecule that is made of these 2 chains, 2 zeta and 2 epsilon chains. Notice that simultaneously, you also have expression of alpha, and the other one is gamma. In the fetal stage, you have production of alpha and gamma. Now, in the fetal stage, you also have expression of the beta globin gene. So you have beta chain starting to appear in the fetal stage, but then at birth, what you have is mainly expression of alpha and gamma, and you do have expression of considerable amount of beta as well. So you have alpha and beta. Now notice something that is really important. So consider this point for hemoglobinopathies, that the alpha gene is expressed early on and it continues throughout life. But for the beta and gamma starts in the fetal stage, but then it drops in the adult stage, and beta starts in the fetal stage and it goes up in the adult stage. Right here. So we have different forms of globin proteins being expressed. We have another one that is expressed, which is globin delta. It is expressed early on after birth, but this person is really minimal. It's insignificant. So you have different forms of hemoglobin molecules. In the embryonic stage, we have a hemoglobin molecule known as GOER1, HBE. E stands for embryonic, GOER1, which is made of 2 zeta and 2 epsilon chains. That's the predominant hemoglobin that exists in the embryonic stage. But you have also small amount of other hemoglobin molecules known as the following. You have HBE GOER2, which is made of alpha 2, epsilon 2. You have HBE Portland 1, which is basically zeta 2, gamma 2. And you have HBE Portland 2, that is zeta 2, beta 2. Now remember, right here in the embryonic stage, production of zeta and epsilon is from the yolk sac. So right here at this stage, you have production of some beta, gamma, alpha, and so you have the expression of all of them except delta. And expression in the fetal stage is from the liver. The main source of globin proteins is liver, a little from spleen later on in the fetal stage, and then bone marrow from the adult stage. So in the embryonic stage, you have almost all forms of hemoglobin molecules. In the fetal stage, now you have a transition into the alpha and gamma. So this is the HbF. F stands for beta. So you have alpha 2, gamma 2. Okay? Alpha continues throughout life. So that's the meaning of fetal hemoglobin. Okay? In the adult stage, on the other hand, now we have a switch from gamma to beta. So the predominant hemoglobin is what is known as HbA1, which is made of alpha 2, beta 2. Now you do have another hemoglobin known as HbA2, which is basically alpha 2, delta 2. And you have very little amount of the other hemoglobins, of the HbF and HbA2. So mainly, just to review quickly, in the embryonic stage, it's mainly GOER1, which is zeta 2, epsilon 2. You do have other forms of hemoglobin molecules, like the alpha 2, epsilon 2, GOER2. But you have other ones having zeta 2, gamma 2, and zeta 2, beta 2. In the fetal stage, it's mainly HbF, which is basically alpha 2, gamma 2. In the adult stage, you have alpha 2, beta 2, red and blue together. You have alpha 2, gamma 2, and you have alpha 2, delta 2 as well. Alpha 2, beta 2 is known as HbA1. HbA2 is alpha 2, delta 2. HbF is alpha 2, gamma 2. Okay? These are really important for you to know for hemoglobinopathies, regulation, and what comes after. I'll stop here. Tomorrow we'll start with regulation.